Todd McDevitt, let's see, bachelor's degree from Duke University. Was it a double major, biomedical engineering and
electrical
engineering? And occasionally he sees an electron, but went on to Seattle to get his PhD at the University of Washington, working with Pat Staten and Alan Hoffman, and then did a postdoc with Chuck Murray, who's on the clinical side, but an academic clinician doing serious research, and actually, it's the Stem Cell Institute here now.
Yep.
So, and then came here in 2004, and his academic appointments in biomedical engineering, but very much a member of IBB, and I'm thinking, Todd, that maybe we ought to get going, even though there are a few people still straggling in.
That's fine with me.
So, and for those that are straggling in, you may have to sit at the front. There are plenty of chairs up front. So.
Thanks, Bob. And there are plenty of seats up here. So, it's a real pleasure for me to have a chance to give this lecture here. For one, this is the type of thing that, and I think you've heard this from many of the previous speakers who have had this opportunity to give a Breakfast Club seminar as well. We often travel and give these types of talks, so we get to explain the story to a lot of external folks. It's rare that we get a chance, perhaps, to go at length. I won't go too much at length, but I'll go at some length about what our labs do, and in particular, try and highlight some of the different aspects today of what my lab in particular is focusing on, which I've identified or titled as Engineering Stem Cell Technologies. So let me give you a little sense of what do I mean by that and where I think this fits within the context of the field. I'm sure that many of you are familiar at least with the term and at least the idea of what is a stem cell. So I'm not going to spend a lot of time on that. I'm also, anyone who was fortunate enough to hear Fiona Watt speak last week, I think gave, as I told her afterwards, perhaps one of the most concise, most efficient overviews of the entire field that I have yet to see in terms of what are the major issues and all of the different types of research that people are approaching. And so within that landscape, and much of what Fiona spoke about was the amazing discoveries that we've seen in cell biology and the history at which this has occurred over the last really 40 years, but in particular in the last 10 to 20 years where we've seen it really explode. And what that has really involved has been really the ability to think about new ways to derive, identify, and obtain these cells, which has allowed us then to be able to study properties of these and what are the functional characteristics of stem cells. Not only their ability to self-renew and propagate and form more themselves, but also a key feature which makes them very interesting to a number of researchers, in particular our lab, is the ability of these cells to differentiate. And when you start to think about the potential to transform a cell into a variety of different cell types, then it opens up a plethora of different types of applications that you can begin to envision as well. Many of these can be on the diagnostic side, new in vitro platforms for both drug discovery as well as new types of perhaps as yet unidentified types of diagnostic tools, simply due to the fact that we have not yet had some of these cell sources before. So it's new types of assays in that area. And one of the areas which I'll talk about a bit about as well with regard to is with regenerative medicine and the idea of actually being able to use these cells and really start to think about mechanistically how do we use these cells appropriately to actually begin to move towards repairing tissues, which is oftentimes one of the very high-profile advertised goals of this field. So why aren't we doing this yet? That should be the question that maybe some of you are asking, and certainly the question that I was asking as I started to enter into this field. I've heard about this and I've seen some of the science, and there's great science going on. So why are we not seeing some of the applications develop? What is the chasm that perhaps exists between these two fields or these two aspects of the broader field? And this is then where I started to think about, well, the traditional role of engineering in particular is to basically transform basic scientific discoveries into actually practical and usable and valid applications. And so I think that that's where our lab particularly interfaces particularly well with this, and that is thinking about how can we actually engineer stem cells or develop the engineering systems that are necessary to really be able to transform some of this science into application. And I think this encompasses three main things, and three of the things that my lab is focused around. One is how do we properly control the cells, particularly through control of extracellular environments. How do we do this in a way that's not just practical at the lab bench, that we can have some nice papers, have some systems that work, but think about from the very beginning, how are we going to actually develop systems that will will enable us to be able to scale these up as we move forwards towards some of the biotechnology and the bioprocessing of the cells. And also then, what is the final product going to look like? It's one thing to be able to produce a plate of cells, but a plate of cells is not what a clinician is looking for. It's not necessarily what a patient is looking for. And so we have to think as well about the whole process all the way through, and then start from the beginning with that kind of end goal in mind. And ultimately then, this is how at least I envision that we can perhaps actually translate what at the moment is much hyped and much discussed potential of stem cells into something that will actually be practically useful. So in the current landscape of the field, and this is my brief one-slide summary to try and capture what the main aspects of the research are, there are a number of different types of stem cells, in particular pluripotent stem cells, which my lab is focused on, and there are a number of other progenitor cell types that can be derived either from this cell type or from other types of other cell or tissue sources. These cells can then go on to differentiate into this vast array of cartoony or these cell types depict in this cartoonish fashion here, which could then be relevant and applicable to a variety of different types of, as I said, applications. Some of these could be disease repair models, others could be new platforms for drug discovery. What has created a lot of excitement in the field, and I've depicted in a manner with the arrows moving in an opposite fashion, is the idea that we can actually begin to take cells that exist in this fully differentiated or somatic state and actually begin to reprogram these. And this has really only been achieved in large part due to the identification of the factors that exist within the cells at this early primitive state, as well as some of the cells in this intermediate state. And so by understanding what was going on here, it actually enabled us to be able to at least perform experiments without the mechanisms currently being understood about how this is possible. But now, whereas traditionally these arrows and these types of figures have always been drawn in one linear fashion, they actually move in both ways. And so this creates a host of new questions about also where else do these arrows exist and how easily do cells transition between states. But one of the challenges that has still not been addressed by this is that how do we efficiently go from one cell state to any single one of these? Or how do we create the paths by which we direct cells and enlarge populations of these cells to effectively go from state A to B to C in a more defined, reproducible, and robust type of manner? So one of these approaches then, and this is where a lot of interest has been in the field, although not necessarily particularly a tremendous amount of success yet, has been with regards to directed differentiation. And this is the idea that we can a priori know what the phenotype is that we're targeting and be able to create the path of the route by which we can get there. If we are effective at that, and that includes, as I depicted here, both the promotion of signaling pathways of different phenotypes or progenitor states, as well as inhibition of others, to really effectively get to homogeneous populations, then the second part is to begin to think about, well, how are we going to do this, as I said, in a way that's large scale enough that it'll actually produce large batches. One of the things from a quality control perspective you wouldn't want is to produce only a few of these multiple times and have to do it in an iterative fashion, because when you get to the quality control stage, you'd essentially have to use all of your product to validate what you had, at which point you now have to start over again. So thinking about how we can do this in a way where we can actually greatly expand upon the success that may be achieved here, and also what are the analytical tools that are necessary to really be able to assess large populations of these cells. They're very, very heterogeneous. There's a lot of noise in these types of systems, and that is a great opportunity, I'll put it as, instead of a great challenge necessarily, for engineering approaches, I think, in particular, to lend themselves to helping with this type of problem. And the last part then is in thinking about the application of the cells. It's fairly direct and has been fairly obvious to some extent that if we can produce any single one or different types of these cells, then the direct application for tissue repair processes would be to inject these cells, either the cells alone, cells with scaffold materials, some type of delivery vehicle. What's been interesting is that while that has been largely the experimental and the preclinical approach that has been attempted, and even some limited clinical studies, what typically happens is not that you get cell replacement from those cells that are transplanted, but that actually there's a transient lifetime to those cells, and then after they're gone, there's some alteration that has occurred due to the presence or the introduction of those cells at the time that they were originally introduced into, say, a site of tissue repair or tissue undergoing active remodeling processes. So this creates an alternative or secondary hypothesis, and that is, what else are the cells doing? If they're not just directly replacing the host cells or endogenous cells that may be lost in the case of a disease or injury, then what role are they serving? And if they're serving a role as more of a complex drug delivery vehicle, can we hijack this or jump on this approach and start to think about, instead of using the cells themselves as a therapy, using the molecules that the cells are producing as a potential therapy? So this is largely, and putting into this context, is kind of where the three aspects of where my lab are primarily focused. And these are three areas which are also interrelated with one another, so I try to depict that in this manner here. The first is in how we can begin to try and engineer and control the stem cell microenvironment with the objective of being to control the differentiation of the cells. Then being able to use some of the tools and technologies developed even at a perhaps micro scale, but in a manner that we can actually readily incorporate these into bioprocessing methods so that we can actually start to greatly expand upon the numbers of cells and do this in a way in which we can actually assess what is going on. And then if we can do this in large enough scale, can we begin to use the cells, as I said, as a way to basically produce molecular therapies, which is, as I said, a different or alternative type approach to what was initially or originally hypothesized for the application of the cells, at least with regards to regenerative medicine. And within this context as well, if we begin to think about what the cells are making, this actually can give us new insights into what are some of the design criteria that we can actually put back into designing some of these microenvironments. So what I want to go through is highlight some of our research and our aspects in each of these three areas and give you a sense of how where they may have started distinct. We're seeing a lot of overlap now between these different projects, different research areas within our group. So what are some of the cues? What are the ways in which we can begin to poke and prod and direct cells? Well, depicted here is a general schematic from a book chapter we wrote a couple of years back, basically about what are all of the different types of biochemical and biophysical forces that cells, in particular stem cells, may be receptive to receiving signals from. And so there are combinations of both the, as I call it, the cell-cell interactions at the homotypic as well as the heterotypic level that can influence these cells, but there are a variety of extracellular, non-cellular mediated cues, soluble factors, extracellular matrix, as well as mechanical forces, both those that can be physically applied as well as those that may exist within this state between the cell-cell adhesions and even from the matrix mechanics themselves. So there are a variety of different formats then that have been attempted to try and mimic some of these processes when we attempt to culture these cells and study this in some type of mechanistic fashion. And this depicts here the differences between a 3D type of system, which are these cellular aggregates referred to as embryoid bodies, which is one that I'll talk at length about today. There's the 2D system where this can be placed onto a defined or adherent culture substrate in the presence of soluble factors. And then this intermediate, this 2.5D, where you can use co-culture models and stromal cell layers as a way to try and recreate some of the aspects of these naturally occurring or even in some cases artificial stem cell microenvironments. So as I said, the one that I want to focus on today, and it could go through a variety of reasons why, is one of them is the embryoid body. And the things that we think are interesting about this is that this is at least a first step towards engineering of embryonic tissues. You're already working with a system in which the cells have a 3D assembly, but they are undefined. And one of the biggest challenges with this system, at least currently, is that it's extremely hard to control. And you can see that although depicted from this nice review by Gordon Keller, embryoid bodies, although they can temporally recapitulate this expression pattern of pre-gastrulation development, they lack the type of spatial organization that you would typically see in an actual developing embryo. That's not to say that you're trying to develop embryos from these, but in a controlled type of fashion, whereas we can go in and oftentimes dissect a developing mouse embryo and know exactly where the cell and tissue types are positioned with regards to one another. In an embryoid body, this tends to always be a heterogeneous type of event. And you can see that here, whereas we start with populations of embryonic stem cells taken through from the early stages of after they've first formed these aggregates to later stages of differentiation, you can see the heterogeneity that begins to ensue both between just the macroscopic appearance of these different bodies or cell aggregates, as well as when we start to look within the inside, you can see heterogeneities that begin to exist spatially between the outside, the inside, and even within the inside regionally, you see specification and differences with regards to the types of morphologies that emerge. And you can then see this, if you look more specifically with phenotypic markers, you can see that although it's regionally specific, you can find markers for different differentiated cell phenotypes, striated muscle, neuronal or ectodermal derivatives. And you can see that although we see these in some spatially defined or confined region, immediately adjacent to these are cells that lack this marker, suggesting that, again, there's a heterogeneous population. Yet, at some level, there is some microenvironmental control that can actually be used to try and get some level of homogeneity among these cells. The other question that struck me when I looked at these is if all the cells start at a similar state, what is it that they're seeing along the way that we can't take them all towards a more defined phenotype? And in a retrospective fashion, this becomes difficult. So one of the two things I want to talk about with this, first is how can we begin to think about engineering control of these systems, these cellular systems, at an individual aggregate or embryoid body level. The second then is how can we start to think about more macroscopically, because there's heterogeneity within this and there's heterogeneity among these two. And in particular, I'll talk first about just our work with regards to how can we deliver morphogens, or some of the differentiation factors that actually can strongly influence the differentiation or phenotype of these cells. So this is the appearance of an embryoid body from the exterior. You can see that it actually has this epithelial layer that has formed on the outside, whereas when we actually begin to cut these open or dissect and look inside of these, and this again was by fragmenting these prior to SEM analysis, you can see that the inside morphology is much different. In a material science sense, you might describe this as more porous, as opposed to something in this which doesn't look very permeable. And you can actually see that right along this edge, as it's been described by other groups as well, there's a shell-like formation that occurs, both due to the cell-cell adhesions as well as extracellular matrix basement membrane deposition that occurs between this outer cell layer and the inner mass of cells. And so looking at this even structurally, we began to ask questions about, well, if things, normally when cells or tissues form this type of epithelial layer, it's to serve as a barrier function. If that's the case in the context of these embryoid bodies, then one of the challenges could be if there's a barrier at the very outside of this, how do you deliver factors effectively to the entire mass of cells? So if we did this, and this is just first with some work with Cell Tracker Red, so this is a small molecule that is taken up at least in monolayer where diffusional transport is not a limitation by an entire population of cells very readily. What we see is that as we do some depth profiling by confocal analysis, whereas the cells on the outside are efficiently labeled with this small molecule, as you start to go through the depth of this, and this is just without the counterstain, but there are cells on the inside here as well, you begin to see that it really is only taken up by the cells on the periphery of this embryoid body. So this highlights that there actually is an effective limitation or barrier to even very small hydrophobic molecule transport inside of this aggregate. And we can quantify that as well as the overall percentage of cells that are labeled, and you see that it's a fairly small fraction or only a fraction of the entire population of cells. So we wanted to ask a fairly simple, in one sense, question, and that was, what if we can change the mode of signaling? And that is, instead of presenting cells or signals to the cells from the outside, which has been the standard manner in which at least most differentiation studies have been performed, could we embed some of the signals, and can we do this in a way in which we can actually control not only the spatial distribution of these, but also the kinetics or the presentation of these morphogenic factors? And so to develop this is I'm going to highlight work of a number of people, and I'm going to acknowledge them at the end of this. The first was to be able to develop a manner in which we could actually incorporate particles inside of embryoid bodies, simply because this had not been explored before. And so using some culture conditions that we had established in our lab, and I'll talk also about some of those in addition to this, we could see that we could actually control this by both the mixed ratio of between our particles and cells at the very outset of this embryoid body formation. We could also do this as some of the mixing conditions that we use. So at lower mixing conditions, we could again get higher levels of incorporation. What this then enabled us to do, and then take a look at some of this, is how are these particles distributed? Not are they just at the outside of this. And so again, with our soluble delivery control, you can see you get this hemispherical appearance. And due to the size of these, with at least standard confocal microscopy, we can typically only image about halfway through most of our embryoid bodies. Whereas in this field, you can see this punctate appearance is actually a number of the microparticles that have been loaded with Cell Tracker Red prior to their diffusional release. And you can see that it's actually distributed throughout the entire mass of these cells, not just coating the outside. If you watch these over time, you can see that they go from this initial punctate appearance, where the dye is, for the most part, contained within the microparticles. These microparticles we're using in this particular example are actually made from PLGA, so they'll readily degrade in aqueous conditions. And as you can see over the course of 10 days, matching with some of the in vitro release profile work we had done prior to these studies, you can see that you actually start to get diffusive release and robust labeling of the cells throughout the entire mass of the spheroid. So this suggested to us that we could get very effective and efficient presentation, or more effective presentation of at least small molecules to the differentiating cells. So one of the things then was, well, what do we want to study with this? We didn't intend to set out and study the effects or the differentiation effects of Cell Tracker Red, although as we've come to learn and appreciate, these cells are sensitive to almost any perturbation you may introduce into their environment. So we actually decided instead to look at a morphogen, or one type of morphogenic agent, retinoic acid, which had been relatively well studied and well published in the literature. And our thought was at least we would have some point of comparison that we could make depending upon what the results of our studies might yield. It's also interesting because the role of retinoic acid has not been clearly defined with regards to its differentiation effects. It's largely thought to be a neuroectoderm or a neuroectoderm-inducing factor. However, you can go through the literature and find a number of examples for differentiation to other phenotypes, including cardiomyogenic. And oftentimes this is attributed to the timing as well as the amount of the retinoic acid and when it's presented to the cells. What hasn't been studied in this context is how it's being presented to the cells, whether it's from the inside or from the outside. So we set out to do this and this is from some of our results. This is after 10 days of differentiation, where we're comparing the native or untreated embryoid bodies, the soluble retinoic acid treated, and then the empty microparticles. So this is a vehicle control group, as well as our retinoic acid releasing microspheres. And what you can clearly see, and it jumped out to us, is that there's a clear morphological difference, at least at the from the beginning of this analysis. And you can see some remnants of it that is suggesting that the presence of the material alone, in particular a degrading material, may have some effects on the differentiation. But what was striking was really when you're releasing retinoic acid from these, you get this much more unique or defined phenotype. And so the secondary question with this was then, what are we getting? And so we initially spent quite a bit of time trying to analyze these and guessing what we thought the phenotype was, and we were always wrong. And so we finally gave up, and with the help of some of our collaborators, Nathan and John McDonald, we actually went to microarray analysis, and I'm very glad, and I think Rich is very glad we did, because this kind of cleared up the whole story for us and got things rolling again pretty efficiently. And that had to do a lot with the fact that we knew that we really only had 2 cell types there. And so when you compare this versus the other groups, in this case one of the groups we compared against were the unloaded or the vehicle control, so you could actually study the effects of the RA release, you could actually see quite distinctly what by the gene expression profile, get a sense of what are the cell phenotypes that were present. And what was interesting to us was that these were not phenotypes that we expected to see. And as we went through and characterized this, and I'm going to just go quickly through some of these, we could see that not only did it give us markers we could identify, in particular markers that were indicative of primitive ectoderm as well as primitive endoderm differentiation of these embryoid bodies. What it also enabled us to do was be able to then go back and with these types of markers, in this case OCT4, which is a pluripotent marker for the embryonic stem cells, but also the epiblast cells. We could see that these are also spatially organized just by presenting the retinoic acid in a different manner than what we had seen with the soluble release. And more striking even than that, perhaps, when we took a look at these and saw the differences in the ultrastructure, again, an indication of the phenotype of these cells, comparing this back to what I showed at the beginning of the untreated or native types of embryoid bodies, you see a very distinctly different organization and structure of the cell aggregates. You see the pseudostratified layer of epithelial that is completely encompassed by this additional epithelial layer that's just completely coated in microvilli. So this looked nothing like what we had normally seen as well. But all of these phenotypes did actually match well with what has been described for actually the early epiblast primitive streak fate of mouse embryos at a stage of roughly E.675. So we had both the gene expression information, the spatial organization of these, and as well as these other morphological markers that all suggested that we were getting something that was very distinct and different. And also interesting to us, this has never been reported, at least that we can find, to be the role of retinoic acid in early embryonic development. And so this nice review actually that just came out from Elizabeth Robertson, who's a very famous developmental biologist who studies gastrulation processes, actually highlights that actually what we are seeing in a completely in vitro system, and this is only with the release of a single morphogen at this point, we can actually get a phenotype of these that's actually been well described and actually looks much like this primitive streak formation that you begin to see at this comparable stage of actually embryonic development. So what it does suggest is that we can actually, through by controlling release or presentation of morphogens, you can get a much more organized type of structure and a much more homogeneous appearance of the cells. So this was very encouraging to us, and as I said, this was with one release profile, one type of material, and one type of morphogen. And so we were excited to then start to push this forward. And so in some subsequent work that we've been doing, and this is some time lapse over the course of 24 hours, we actually have developed, or haven't developed, we've taken advantage of a system that one of our collaborators has actually developed, in which you can actually begin to form embryonic bodies at extremely high density. And this is with even more homogeneity than we had originally seen. And the idea with this is that simply by using a templated surface, you can begin to centrifuge or force these aggregates of the cells to form in these micro-pitted wells. And you can see that the cells stay within this. They move around quite a bit over the 24 hours. And by the end of this period, what you see is that although they're kind of distributed throughout and they tend to align because these are the edges of the pyramids, what you see is over 24 hours, they start to form these primitive or these initial cell aggregates. The nice part is that you can easily rinse these back out of this system and retrieve these in a pretty easy fashion. So we had a question then, which was, well, if you can do this with cells, can you not do it with materials as well? And one of the ideas with this was, could we use this as a way to not only improve the effective control of particles per embryoid body that's formed, can we also do this to be able to improve or expand the number of types of materials that we had been looking at? And the answer with both of those is yes, we can. We actually get a much more, compared back to our initial system where we were simply mixing particles with cells, what you can see is that we do get a fairly homogeneous population with just our rotary culture system that we've been working on. But when you actually start to use the agar well as they're described, and these are commercially sold now, you can actually get incorporation within all the embryo bodies. It's much more uniform overall. And we can begin to incorporate materials such as agarose and even PLJ that's not coated. So in our initial studies, we had to make those materials adhesive before we could actually get them to be incorporated. In this case, you can now actually essentially force any of these types of materials that appears in everything that we've been able to test so far, we've been able to effectively incorporate. So when you can do that, you can start to think of a whole new realm and spectrum of the kind of release kinetics and the types of morphogen presentation that you can make within this cell spheroid, and that's actually exactly where we're headed in the future. So what we were looking at this is at least one example of a platform system we can begin to build upon. The idea with this then is we can actually begin to think about what are all the different types of properties and things we'd like to engineer into the materials so that we could actually introduce these into the environments and ask very direct and specific questions about the roles of various types of morphogens and even potentially the inhibition of endogenous morphogens within this context. And so we're now, as I said, building upon this work and asking some of these questions. As we've been doing this, one of the things we've been, as I said, thinking about is not just how can we control the microenvironment of the individual cell spheroid for, as one example, how can we begin to think about, as we want to scale this up, what else is going to be important? So there are a number of things that are typically used, again, to try and control the overall differentiation of the population. Many of these are the composition of the media, other environmental factors, pH, oxygen levels, which can be readily controlled with some of the existing and even more, and some of the bioreactor systems that are currently under development. One of the other focus has been on the size of these aggregates, and we've seen variation with this, as I'll show in a second, with some other means. But one of the things that, for example, that agar well has been commercialized is because it can so effectively control that, so that it really helps eliminate a large amount of heterogeneity in the system when you can effectively control at the single cell level the number of cells per those aggregates. And that in turn affects some of the cell signaling and differentiation that you see. But one of the things we're also interested in is that as you actually start to think about taking these from bench scale systems to larger volume reactor systems, you also typically introduce some mechanical forces, at least in the form of hydrodynamic forces. So although most studies on embryonic stem cell differentiation have been done at a level at which you have these small static cultures, either small individual volumes or small petri dish volumes, as the proposed methods go forward to bioreactor systems, you're talking about a scale up again, at least in terms of volume, of 2 to 3, if not more. But the other thing that gets introduced typically in these systems are the hydrodynamic forces that are imposed by the mixing or the stirred conditions of most of these types of bioreactors. And if you were to then try and go through and analyze the various combinations or the various applications of these types of hydrodynamic forces, you'd find it's quite limiting in the sense that these large volumes occupy large amounts of incubator space, and so it's not a very high throughput platform. So one of the questions we had about this is there a way that we can begin to at least simply introduce some hydrodynamic forces at a much smaller scale so we could actually use this as a system upon which we could examine what effects this may or may not have on differentiation. And so to do this, we started with, as I said, our fancy bioreactor system, which are simply these horizontal rotary orbital shaker plates that can be actually, in collaboration with with a researcher from the University of Louisville, actually we've been able to define, at least to an extent, some of the fluid mechanics in this environment. And this has enabled us to show that this is a relatively low shear, fairly well-defined type of environment that the cells are consistently exposed to over the course of, say, 7 to 14 days of differentiation. But one of the things we found is that in contrast to some of these smaller volume techniques that are commonly used, the hanging drop where you suspend small numbers of cells in individual drops from the lid of a petri dish, and here, in this case, we've knocked them off, you can get fairly good homogeneity of the population, but it's not a high throughput method by any means, and it's actually, there's no real way to really be able to scale this up effectively. In static suspension, what you can see is that you can get initial formation of these aggregates, but they'll quickly begin to agglomerate with one another. they'll also begin to then form these secondary embryoid bodies, as they might be called, and you get this very heterogeneous environment. And so the differentiation within any single one of these aggregates or in the overall population is extremely hard to define. But what we can see is from even the very early experiments is that by just imparting this simple and continuous fluid motion, what we can actually see is that we get a much more homogeneous appearance of the cells. And we went on and also did some analysis of this in our first paper to describe the fact that you actually enhance the efficiency of formation, the overall yield of the embryoid bodies, as well as the number of cells, and also that you can inhibit this agglomeration event. And this is something that we were then interested in because it had a lot of practical applications. But the other thing that was interesting was that in some of these early studies where we were actually at the time comparing differences in differentiation, in particular, for example, cardiomyogenic differentiation, you could see that hanging drops, which produce a cardiomyogenic phenotype with fairly good reproducibility on a per embryoid body basis, the static culture system doesn't lend itself to that, and the frequency of this event is oftentimes much lower. But what we could see is that by simply plating individual bodies and counting the frequency of this event, that the hanging or the rotary culture system, or the embryoid bodies produced by the rotary culture system, actually tend to be an intermediate that was significantly greater than the rotary system, but not quite as good as the hanging drop. So something that looked similar in morphology was actually behaving similar. But this is a very, I would say, non-definitive type of assay, or at least I always say it's a non-definitive assay. This is a good screening tool. So Carolyn went through and did a significant amount of work to actually characterize this in more detail. First, looking at the gene expression profile of these cells, in particular looking at some of the cardiac transcription factors, as well as some of the gene expression for some of the sarcomeric muscle genes. And in almost all of these cases, we saw significant differences, or at least an enhancement in our rotary group, suggesting again that what we were seeing, one of the reasons we might be seeing the frequency increase in that spontaneous contractility was due to the fact that there was actually some endogenous increase in the gene expression for cardiomyogenic factors. But that again is a population-based type of analysis. So Carolyn went further with this and also could demonstrate that there was an increase, although not significant at least, due to some of the heterogeneity that exists in the system. But we could see that there was a consistent increase we saw even at the cellular level. So this suggests again that this rather simple change in the initial start of our culture system was having an effect on the population of cells that emerged, or at least one phenotype of the population. So this caused us to go back and look a little bit more closely at what we were doing and perhaps what effects it might be having. And so we went through, and Carolyn actually then went through with all of these and started to actually vary the rotary orbital speed she was using as a means to try and control the hydrodynamic forces. And this also then produced what we could clearly see were differences in the formation kinetics as well as the size and yield of these embryoid bodies. And so you can see that here is that at some of the slower speeds you get a larger size aggregate. When you start with the same number of cells then, and as we quantified, you get a lower overall yield, at least of individual aggregates. But the total cell balance or material balance for the cells is equal. And then over time you can see, with the change in speed, you can actually see that you can begin to modulate this size. As I mentioned a little while ago, modulating size is interesting because that's already been shown in other types of systems as well, that might be a manner in which you could actually begin to control the differentiated phenotype of the cells. And so we then took some of our different rotary conditions at which we saw some of these differences in at least the morphology and the yield of the cells, and went further with this and have done a bunch of differentiation analysis. So I just want to highlight a couple of things. One of them was when we did this, I should call this really semi-global or low-density gene array analysis with these quantitative real-time PCR arrays from SA Biosciences. There were a couple of things that struck us when we compared our static groups to some of these different rotary groups. The first was that there were gene expression differences in some of the genes that were on these arrays, and these are commercially produced, so these are actually non-biased in the sense that whatever was on the array, well, we got whatever we purchased, is what I would say. We didn't get to actually a priori choose what was going to be on these. But it was interesting because these would not necessarily have been genes we would have chosen, but you do see clear and distinct and significant differences between cells that are being differentiated under static conditions, so the traditional culture format, and those that are actually being cultured under these different rotary hydrodynamic conditions. The second thing that we noticed that was perhaps a little more subtle but also interesting was that there were a number of genes where we actually saw differential gene regulation as well, suggesting that it's not just whether there's mixing or not, but actually the levels of mixing can have some effect on the gene expression profile. And that was at one of the time points when we actually started to then look for and ask questions about, is this changing the kinetics of differentiation or the overall phenotype or percentage of the different phenotypes that we get? One of the things we could see is that if we looked at pluripotent markers such as OCT4, NANOG, and some of the early mesodermal markers, such as brachyury T, you see that there were no differences between our different rotary groups, suggesting that we're not just simply inhibiting or delaying differentiation in some manner, that all the cells are as a population proceeding similarly. The difference was when we actually started to look at what are some of the different, at least enrichment of some of the phenotypes we could see. And looking for markers such as nestin, for the most part, a fairly good ectodermal marker, and alpha-fetoprotein GATA4, which is our pretty definitive markers for the endoderm and mesendoderm, you could see that we did see differences, significant differences in these populations, such that you could actually begin to use this not necessarily to get completely directed differentiation of populations, but you could begin to actually push the cells in slightly different directions. And this is completely under the same culture conditions or soluble media, FPS containing media for all of these studies. So we're not actually changing any growth factors or anything like that here, at least the ones that we add. One of the limitations from these, and one of the things we're now working on further to really assess this and what is the effect of these hydrodynamic forces, comes from some additional follow-up work we're doing on this, and that is, can we normalize for the initial formation of the embryoid body and take that out at least as one of the parameters. And so from some of our recent work, we can show that using those agar wells that I presented earlier that we could use for the microparticle formulation or incorporation, we can actually begin to normalize the population of these, subject the populations to different rotary orbital conditions, and then really begin to assess what are the effects of the different hydrodynamic forces they may be exposed to during different periods of culture time. So this brings up a potentially new and subtle and yet undescribed concept that we had come across, and that is the idea that perhaps hydrodynamic conditioning could be used as another, or at least as an adjuvant, to be able to control or direct the differentiation of the cells. And I say this because, you know, this is interesting in the context of, as I said earlier, the cells appear to be sensitive to any kind of perturbation. And so I think this highlights that as you change and you think that just because you've added the same growth factors, you've delivered them at the same time, and you're only focusing perhaps on soluble, which may still be the predominant cue to the cells, when you start to actually even make a subtle change going from a static environment to the hydrodynamic, or something where there's mixing involved, that also adds another level of complexity, and it can actually also needs to be taken into account, both in the design of the system as well as in the analysis. So what I want to just conclude with very briefly is just some of our work then in what else are we looking to do with these cells. As I alluded to, if we can control the differentiated phenotype, that will enable us to perhaps then think about some of these types of applications for regenerative medicine as well as diagnostic technologies. There's a secondary, as I said, though, hypothesis, and that is what else are the cells capable of doing, or in this case I should say producing. And can we again use this or begin to think about ways in which we can actually intentionally use the cells for this type of purpose. So it requires a couple of things, and the first of these highlighted here is that what are the cells actually expressing, and how does this change as a function of the differentiated state of the cells. And so again, we've used some of these low-density array platforms where we can actually do some temporal profiling. And in this case, again, looking at between days 4, 7, 10, and 14 of differentiation, so early differentiation. Highlighted here in this array are, in particular, extracellular matrix proteins, some of the adhesive receptors for those proteins, as well as some of the MMPs. And then also in a second or parallel array, there's actually ones where you can look at the growth factors and cytokines. And what's striking about this is that you can clearly see patterns of expression that cluster together. The nice part, too, is that the patterns that we see between our replicates actually also cluster together temporally. So there is a pretty controlled or sequence or progression to these types of events. And these are the actual morphogens, or at least I should say some of the factors that the cells are producing locally within this microenvironment that they're endogenously making. And when we take a look at some of these, then one of the things you can begin to do is think about, well, what are the, in this case, early cues that are associated initially or correlated with this differentiated event of this embryonic environment. And we can see that there are a number of molecules, some very interesting, some that we may have speculated on, others that we would not have, again, necessarily picked or focused on initially, that are increasingly expressed with differentiation. Similarly, there are a number of growth factors that are also associated with the promotion or differentiation of the phenotype of these cells. One of the other things that's also interesting, there is a subset within these of growth factors that are known to be associated with the pluripotent phenotype of the cells. And we see that although those are expressed initially, they tend to also decrease over time. And so two of the things we can then begin to get insights about from this system, one, what are potentially the factors that promote differentiation? Could we engineer these into our system, perhaps through microparticle or other types of means? And also, what are the other types of cues? These have been studied and well known, these 4 factors in particular. What are the other factors with regards to extracellular matrix and other proteins that are not as well known that perhaps also are responsible for these cells remaining in an undifferentiated or self-renewing state? So we're now using this information, looking at this, and this is just some of the gene expression, to actually start to try and do some additional analysis, and also for 2 purposes. One, to better understand what are these systems and what are the potency or the importance of the relative systems or networks that may be involved in directing differentiation of the cells, as well as what are other putative targets that we should be looking for, both with regards to the extracellular molecules or factors that are being produced by the cells, as well as some of the downstream signaling pathways that may be involved in this type of process. And much to our pleasure, as I'll show in a second, highlighting one of these, which is the interaction of Versacan and hyaluronan in particular, which appears to play a role in these early, or it's one of the earliest networks that appears to emerge within the system. This is one of the ones we had happened to take a look at, and I'll show some data for that in a second. So looking then at the gene expression, we can then actually confirm this by immunofluorescence staining, as well as additional proteomic types of techniques. And you can see that there are whereas the cells start largely as a cell aggregate, so there's very little matrix that's present in the early time points. Increasingly, you can stain for and see the expression of various matrix molecules, which we could link back to some of the gene expression data. And also, you can see from this the IF staining also gives you some semblance of the spatial organization, or where are these molecules located or associated within the differentiated population. And one of the ones that we touched on, and I just want to highlight for a second then, is hyaluronan Versacan, which is actually a glycosaminoglycan. So hyaluronan we would never have picked up from a gene array because there's not a gene for it. There are genes for some of the Haas enzymes, as well as Versacan, which is a proteoglycan that is closely associated with hyaluronan. It has a HA binding domain. So in some work we did in collaboration with Tom White and Marcia Raleigh with Shreya Shrukler, former undergrad from the lab, and Reka Nair, one of the things we looked at was actually you could see that these were co-localized. So where we saw expression of HA, we also saw the expression of Versacan. And we saw that it was progressively increasing within these embryoid bodies as they differentiated. The other interesting thing is that as we began to try and say, well, what cell types or phenotypes are associated with where these cells or where the matrix is being produced locally, we could actually see, for example, that with an epithelial marker such as pancytokeratin, which appeared to be in the areas that were completely absent of Versacan, whereas in contrast, an early mesenchymal or mesodermal marker such as encadherin appeared to be enriched in some of the regions where we were seeing Versacan being expressed. So what this also then highlights is that these endogenous molecules being expressed by the cells and where they're being expressed actually could then be affecting the differentiated fate of these different cell populations. We've also, in some work that we've been doing in collaboration with Barb Boyne and Bob Goldberg and Zvi Schwartz, also been taking a look at what else are the functional characteristics of these matrices, and with regard to at least one example where upon addition of beta-glycerol phosphate, we can begin to see that some of these embryoid bodies then will begin to actually mineralize these matrices. So this is some functional ability to look potentially at the osteogenic potential of these cells, which we're also doing in parallel, and then think again about the utility of these matrices to actually potentially stimulate some type of at least embryonic-like mineralization. And then going through and looking at this with a field of these under stereoscope, you can see that very few mineralizing by micro-CT, whereas when you look at a population that's been treated, you can see that almost all of these and throughout the volume of these appear to be mineralizing. So we're looking at this as a way now, like I said, to kind of look towards the function of some of these matrix components. One of the other things then that I just want to conclude with is, well, how are we going to actually use the matrix, or what is one of the ways in which we can actually begin to think about intentionally deriving matrix-based products or acellular approaches to this? And one of the ways this has been done, at least for a number of somatic tissues, has been through the ability to decellularize existing tissues to remove the cellular component and obtain or extract that extracellular component, which is largely composed of the extracellular matrix and a lot of the growth factors that are associated with that. One thing that may be interesting in particular, and one of the things that's motivating this work, is that, as many of you are probably aware, at least in mammalian species, as we develop, or as we develop from early embryo through fetal stages of gestation to all the way through to adulthood, in general, our regenerative capacity decreases over time. And so what this suggests, or some of the questions that have been raised, is why is that? And in this case, in particular, where we're looking at an embryonic morphogenic event, are there elements of an embryonic environment that perhaps contribute to a fetal-like wound healing response, which is in a scarless fashion, compared to the adult wound healing formation, which, as many of you know, typically leads to scar formation and a loss of tissue function. So in a series of work that we've done, we have taken embryoid bodies and looked at various ways in which we can begin to decellularize these using either a variety of different types of solvent and enzymatic-based approaches. Focusing then on one of these, which appeared to be very efficacious in our hands, we could actually examine the ability to do this on different stages of the embryoid bodies. So this actually gives us temporal control to extract perhaps the matrices at different stages of differentiation, as well as in combination with directed differentiation approaches, perhaps towards different types of phenotypes. And one of the important things that we've seen as well is that many of those matrix molecules that we saw present before we decellularized are still present after the decellularization process. We've also looked at alternative formulation approaches with this for a number of means. I won't get into the details, but have shown that we can also do this through physical or mechanical perturbation methods in addition to some of the solvent-based methods we initially developed. And this has then also enabled us to begin to start to ask questions, and this is leading into more of the biological types of studies that we now want to be able to do, which is seeding cells back onto these matrices. Somatic cells in particular, what types of unique responses might you see from somatic cell types, and especially those that may play a role in wound healing, compared to a normal type of or adult-derived types of tissue matrix. So this is, as I said, we've been looking then at the start of this, and this is the idea of basically being able to create stem cell-derived matrices by having a controlled differentiation system where we can control potentially the specificity as well as the timing or the maturation state of the cells. And now we're very focused on how can we begin to scale this up in a manner, as I talked about, scalability is important. And also, what are the formulations that we can actually take this and deliver it in a form that will be useful for at least our in vitro studies, but also as a potential clinical therapy as one potential approach. So this is, as I said then, I just want to highlight how are these different elements interchangeable, or how are they interacting within the context of our lab. I want to highlight the folks that have done all of this work. I think, again, it's understated, I think, in particular, just how much this group in particular has been able to accomplish in a relatively short time. And I'm extremely lucky on a daily basis to work with this group of people. They challenge me, and it's a lot of fun, at least for me as well as I think for them, to be able to work in the environment that we do. And it's one of the things that I feel very fortunate to be a part of. I think I'd also be remiss if I don't, and I always try to highlight the group as a general, but there are three people in particular I really need to highlight from this, because really the bulk of the work that you've seen have emerged from, and they conveniently all stood next to each other for the picture, which was nice. So Rich, Carolyn, and Reka, who, as I always say, were bold enough to take a chance with a young professor who had no lab, had nothing going at the time, no projects, formally established, and have really been the instigators and the root, the cause of all of what you've seen today. So again, when you look at how a program can expand, and you can link it back to three people in particular, I think that deserves some mention of that. And that has also been extremely important, because then you see all the rest of these other folks that have joined the lab since that first year and have contributed significantly to this as well. So we've had a number of, as I said, the group members that have been involved, lab alumni and undergraduates who have worked in the lab. And unfortunately, I don't have time to go through it, but a tremendous list. When I sat down to go through this and thought of all the people I've been fortunate enough to work with over the last 5 years or so, it's been really a great experience. It's really helped to expand my perspective on the field and the ability and projects and think about what we can do. And I want to highlight both those that are local as well as those who are external to the Georgia Tech Emory community. So stop there and take any questions, and of course thank the funding that has made all of this possible.
So Rich, Carolyn, and Reka basically were foolhardy enough to join your lab.
I wanted to say it nicer than that, but yes. Brave.
Todd, I thought the differences you saw in differentiation with different doses or magnitude of the hydrodynamic environment solution, how does that compare to the physiologic environment? And has anybody looked at different types of environments, cyclic hydrostatic conditions or things like that?
Not so much on the embryoid bodies. I would say because of the fact it's probably the format that they lend themselves to, because they're just a cell spheroid. It doesn't mean it's not possible, though, and I could envision that with the development of mechanical systems that you could actually then try and load these and see what the effects are as well. With regard to the first part, and this brings up a really good point, if you compare this back, and so to be fair, an embryo body is like like a developing embryo, but it's not. So you could make some say it's not exactly the same anyway. But one of the things that's interesting is that I would consider this one of those examples of a non-physiological cue, but perhaps well controlled. And I think what we've been looking at in this one we're interested in is that if you can understand the mechanism by which, we may not ahead of time have an example and say, oh, well, the closest we got to it said, well, embryos are bathed in a fluid-filled environment. Yes, but they don't rotate at the frequencies that these are being applied, and it's not the same kind of structural or fluid mechanic environment at all. But what it does suggest is that if you understand the mechanism by which it is, you can actually perhaps control some of the endogenous signaling. So it may be non-physiological in the sense it doesn't relate directly to developmental processes, but you may be able to stimulate specific developmental processes in a much more controlled way. One of the advantages of that is that typically we do that with growth factor treatments and other things, which at least at current production value is extremely expensive. And one of the things here is that you may be able to, as I said, kind of hijack an existing system, which is they're going to make things on their own. But if you can do it in a controlled way, you might actually be able to use that to your advantage. So that's kind of how we view it at the moment, is that I would say it's a non-physiological.
I have a follow-up question to Bob.
Yep.
Well, right now your attribute seems like one of the working hypotheses that the difference in differentiation is due to hydrodynamic forces, say shear forces, some other forces, because of the identical environment, physiological environment those cells were.
Yep.
However, if you look broader to this problem with the rotated system, because of the non-uniform velocity field, actually your local chemical microenvironment also dramatically different. So even though you may have created the global environment the same in all these tissues, actually local environment in the cell seed, pH, growth factors, concentration can be dramatically different from where the cells are located.
Absolutely, absolutely.
So that's kind of will bring it back to just environment control.
Right. And I think that coming back to that, that's why I think that both are important, I think is what I would say. And that in the one context, I think that to really get at the microenvironmental control, you're going to have to be at a scale or level, microscale, where you're actually controlling that. And I don't think that a hydrodynamic or those types of forces are going to play that role. What's interesting is how what I would describe as sets the baseline. And that is that the baseline may or may not be either permissive or inhibitive for certain phenotypes. And what's interesting with that is then that you can begin to think about, it's just as important to be able to perhaps inhibit certain pathways or certain phenotypes if you're trying to get to a more homogeneous outcome in the end. So the way I envision it is that, it's going to be a combination of technologies that would eventually be necessary. But until we had looked, and perhaps now some others, it was not being taken into account that maybe you should take some of these other factors in as well. The general approach is to look at one factor at a time. And when you do that, you can typically find a difference. And that's how we get papers out. That's fine. But I think when you start to combine these and think about it, it's much more complicated what's going on. And when we make a subtle change like that, it at least needs to go into some of the potential analysis of the cells. Great point.
Great. So following on Andre's question, there's a linearly varying shear stress there. So maybe they're responding to the maximum stress at the periphery, or maybe some average stress. Maybe there's a different metric than RPMs that would illuminate that dependency, and maybe you could create an environment that was more uniform, like a microchannel, to sweep us a wider spectrum or keep it more uniform. Yeah.
So I didn't get too much into future work, but both of the points that you raised, and this gets back to is projects I would say evolve. We didn't appreciate initially that whether or not this was going to have an effect or not, but what we did start to quickly appreciate through reviewers and additional analysis was that it is still a heterogeneous, it's a more defined but it's heterogeneous environment. And so we've actually taken this as motivation to begin to move to more defined systems, to really examine this, as you were saying, to look at perhaps using microfluidics and other types of better controlled systems to ask those types of questions. This system probably doesn't lend itself to it. This system was the motivation, though, to perhaps move to some of those, is what the way we've looked at it. And that's actually exactly what we are doing with some of the additional work. But it's an excellent point.
So I'm not going to follow up on any.
Thank you.
So my question is about the diffusion work you did with the retinoic acid and the cell tracker red. What are the differences in the size or the characteristics of those two molecules? Because, the way I think about it, there are cells on the inside of an embryo body that need things that have to come from the outside. So
Yeah.
what are the properties of those two molecules that maybe are different?
So at least molecular weight size, they're roughly the same, under 1,000 Daltons. One of the things that'd be interesting with that is that, you know, the ES cells of this, they are positive for gap junctions. They form gap junctions with one another. So small molecules like that could also, in theory, transit between the cells through the gap junctions. Now, whether the gap junctions form differently at the exterior versus the interior would be another limiting factor. But in a molecular weight size, they're roughly the same. They're both, I could generally call them hydrophobic. And so we've actually, though, that's using one of those. We've looked at these with other and start to vary the molecular weight as well as some of the other, you know, the PI and other types of things of the molecules to try and get a sense of how limiting this is. I think with that example, or why I use that as the example, if a small hydrophobic molecule is not easily going to traverse across the first layer or multiple layers, then what hope or chances, or what's the likelihood that larger morphogens, growth factors, are going to be able to freely pass across? And this is also a function, I didn't get into some of the details of it, it's a function of as they differentiate. So the question too is as what's happening. Well, I showed the one image of this, or the quantification of this, that as they differentiate, how that potentially changes. So is it an actual, the dynamic versus passive kind of effects of the environment? So we're looking at that now to try and better assess what potential molecules. Certainly with small enough size aggregates, it may or may not be a problem. That's one of the other questions. And so we've looked at that as well. What are the effects of size?
First of all, it's great that you're already thinking about issues like scalability and bioprocessing. I think it is very important for translation, especially as engineers. We should not forget that. Also, you saw these interesting differences between the static versus rotary culture. And it could be shear stress, but have you thought about the mechanism and possible other mass transport issues or combinations?
Yeah. So that's exactly where we're trying to head with that now. And it's a combination, like I said, of those. And so trying to find better ways by which we can actually analyze mass transport. One of our collaborations, it's an early collaboration, so I didn't want to get it, you know, I have one picture, I don't have results yet, so I figured it would also raise more questions. So with Ari Glazer, and the whole question there is based around what are the transport properties of these, and can we define them first and then be able to, if we're going to actually try and overcome them. So that's one of the ways in which we're beginning to look at, as I said, some of the new projects that have emerged based on some of these principles. But, sorry, first part of the question was, I'm going back.
First was I was just like complimenting you on your thinking.
Oh, yeah, say that one again. That's all right. No, but yeah, I think the thing is, as I've gone, I use this kind of interchange or interrelated nature. One of the things that's been really interesting to see is how each of the things that challenge one thing that we're looking at in one context actually affects the other. So I'll give the other example. So the particle work. When you start to put particles inside, how do you actually affect the diffusive properties of the, so is putting a particle alone inside enough to alter the transport that now you can actually get things across in a soluble fashion? We didn't think that until you start to see some experimental data and you'll start to question this and look at it. So I think that is what I'd say in general sense, yeah, all of these are related. We have to find ways to be able to dissect them, understand individual roles, but also study them together.
Is there a final question? If not, Todd?
Thanks, Bob. Thank you. All these are